out. I think we are. Hello, everybody. Thank you for joining Lindsay and me today. We are going to talk about San Francisco Bay's tidal wetlands and the native and invasive Spartina species that we have here. Okay, so first, I'm just going to give kind of an overview on our tidal wetlands, and then we'll talk about Spartina in a little bit. So tidal wetlands are a very prominent feature in San Francisco Bay. They're pretty extensive up in the far north and in the far south, where we get the freshwater inputs mostly coming in, as well as Sassoon, where the delta meets the estuary. These are easily noticed, especially when flying out of SFO in Oakland. If the tide is low, you can see a nice mosaic of the brown mudflats with then green bands of tidal marsh, and then you've got some kind of puzzle pieces of managed wetland ponds. You might also notice it as you're crossing the Dumbarton or San Mateo Bridge, going out of the east side in late summer and fall, because it starts smelling really, really bad. These zones are all really, really important to the health of San Francisco Bay as a whole, as well as to us humans who live, work, and play along its edges. The tidal marsh zone is essentially the upper vegetated portion of mudflats. So it's sandwiched between the marine and terrestrial ecosystems, and it kind of provides the main connection between saline and freshwater systems. These are highly productive ecosystems. They're really rich in biodiversity, and they provide critical habitat for numerous aquatic and terrestrial critters who feed, take shelter, and breed in the marshes. They also are very important for our shorebirds. They act as a daily high tide refuge when the tides come up for shorebirds that at low tide will be feeding on the mudflats. So our tidal marshes also provide a lot of ecosystem services that are really important. They act as prominent carbon sinks, which is a big thing right now as we're trying to combat climate change. They also protect the coastal zones from natural disasters. So they act as a buffer from storm surges, and they help protect shorelines, like urbanized shorelines, from erosion. They're also very important in processing contaminants. So a lot of contaminants that come off of our urban hardscape get filtered through the marshes, so it keeps our bay water cleaner. And so these are all really important, but unfortunately, globally, 25 to 50% of all salt marshes have been lost to human development. Slide. So this is very much the case in San Francisco Bay. So this slide shows the historic and current geographic extents of tidal wetlands within the bay. You can see historically, very collected up in the north, San Pablo Bay, Sassoon Bay, and down in Central and South Bay. So that's where Petaluma River, Sonoma Creek, and Napa Rivers come into San Pablo, where the Sacramento River and American come into Sassoon. And then in Central and South Bay, it's Redwood Creek, Coyote Creek, Old Alameda Creek. This interface is where the tidal marshes come together. Unfortunately, we have lost about 85% of our wetlands in the last 150 or so years. The slide on the right has some different colors going on. The bright green is this current tidal marsh. The kind of middle shade are tidal marshes that have been restored in the last 40 or 50 years. And then the really dark green, those are future restoration projects that are in planning or some level of implementation currently. So we lost about 85% of our wetlands in estuary, all due to human development. A lot of lands were diked and filled for agriculture or for residential and commercial development, and a lot of them were levied off for commercial salt production. With all of this reduction, San Francisco Bay Estuary still contains the largest remaining expanse of tidal marsh on the western seaboard. this estuary is also the second largest in the entire country. So these are, while they've been greatly reduced in scope, they're still extremely productive habitats. They release nutrients and create organic material that supports most of the bay's food web. So this includes aquatic plants and benthic and aquatic invertebrates, which then support the fishes and the birds and mammals and everything from the bottom up. So marshes also provide a lot of protected areas for a bunch of animals for foraging, breeding, nesting, and this includes several endangered species. We'll talk about that in a sec. These habitats are also really important for waterfowl and shorebirds over the winter. This is a lot of their wintering habitat as they come down from breeding in the Arctic. But it's also an incredibly important stopover area for more than a million other shorebirds that migrate through the Pacific Flyway every year. So with all of that, San Francisco supports the highest density and abundance of wintering and migrating shorebirds on the Pacific coast, which has led for it to be designated a wetland of international importance by the Ramsar Wetland Convention and an important bird area by the National Audubon Society. Slide. So we just saw how the amount of tidal marsh habitat has been reduced dramatically in the last 150 years. That has led to some of the species that are dependent on the tidal marsh being threatened or endangered. And these are the three that we encounter the most. We've got the endangered saltmarsh harvest mouse, the endangered California ridgeways rail, formerly California clapper rail, and the threatened California black rail. Slide. So historically, the biggest threats to tidal marshes in the bay has been human development. We've largely curtailed that, but we do still have some future and current threats, mostly surrounding climate change. These involve sea level rise and potential for marsh drowning. That's exacerbated because most of our marshes are right up against hardscape. We've developed everything. It's all freeways and airports and shopping malls. So they don't really have anywhere to migrate up as sea level rises. We also might be encountering more frequent or intense storms, which can increase flooding. It also can increase erosion of those marshes as waves become bigger. So while these are threats to the marshes themselves, these marshes are also protecting the coastal communities that they're adjacent to from the exact same factors. The marshes will mitigate waves more from these bigger storm surges. They protect against flooding, filter pollutants out of the water that's flowing into the bay, reduces erosion on our developed areas while sequestering carbon. And as has become incredibly important over the last couple of years, especially with the pandemic, they provide a lot of open space for recreation. Slide. So there's kind of a lot going on this slide. I get it. But this is a brief little glimpse at kind of the history of tidal marsh conservation in the bay. It mostly started in the early 60s and as a reaction to a huge development plan called the Vigor plan that was being proposed from the 40s to the 60s. Basically, two giant dams were going to be built kind of at the Bay Bridge and the Richmond Bridge and create these really big freshwater lakes with a much smaller estuarine system with a lot more fill and dredging, and it would be a major shift to the setup of our bay. In the early 60s, three women reacted very strongly against it and formed the Save the Bay. Save the Bay's whole vision is to kind of get people on board with realizing that our marshes and our wetlands are important and they should be valued and not just developed. They in turn helped pass some legislation, which formed or established the BCDC, which is San Francisco Bay Conservation and Development Commission. They are a regulatory agency basically to stop degradation of tidal marshes and wetlands. My screen just went all weird. So with that, in the 70s and 80s, there started to be a big movement for wetland restoration. So trying to kind of reclaim these lands that had been developed in some way. The easiest ones to deal with were the levied salt ponds, commercial salt ponds. So those started in the 70s and 80s, and the Coastal Conservancy, the California State Coastal Conservancy, was established in 1976 to kind of shepherd that process, to usher in these restoration projects that are moving forward. Then jump to the 70s, and there was introduction of East Coast Spartina alterniflora into San Francisco Bay. We'll get into a little bit more of the mechanics of that. That East Coast species then hybridized with our native species sometime in the 80s, and it just started running rampant. It started converting a lot of the landscape. People started taking notice. Studies on this curious plant were conducted in the 90s by UC Davis, which is when they identified that that's what this was. This was a hybrid between the East Coast and our Pacific Coast Spartina species. So through that time, up until about 2000, a bunch of the landowners and land management agencies were trying to control the spread of this plant that was taking over their marshes. That's like East Bay Regional Parks, U.S. Fish and Wildlife Service, and they just realized that they couldn't do it on their own. And so that is when the Invasive Spartina project was formed in 2000 to coordinate this regional effort, because nobody was able to really do it on their own. Slide. So this is who we are. And I realized, yes, so the Invasive Spartina project is a project of the State Coastal Conservancy and the U.S. Fish and Wildlife Service. So Coastal Conservancy is our state lead, Fish and Wildlife Service is our federal lead. And it's then CalIPSI, or California Invasive Plant Council, and Olufsen Environmental, or OEI, we're the ones who manage and conduct the work on the ground. So Lindsay and I both work for OEI. I imagine you're probably all pretty familiar with CalIPSI, but if not, they're a wonderful resource. They're a statewide nonprofit that provides a lot of resources for land managers and organizations trying to deal with wildland weeds. If you're not familiar with them, I bet Susan can hook you up. So in addition to these four main groups or organizations, we have about 200 other partners around the bay. Pretty much imagine any property manager or landowner that touches the bay, because any shoreline, whether it's vegetated or riprap, it's all potential Spartina habitat, invasive Spartina habitat. So this includes all bay-fronted cities, counties, local, state, federal agencies, water districts, mosquito abatement districts, and a lot of private landowners. So that's kind of our general tidal marshes and the groups that are involved with tidal marsh restoration and conservation around the bay. I'm going to shift and talk about Spartina itself a little bit. First, we're going to start with our only native Spartina. It's called Pacific cordgrass, or Spartina foliosa. It is, again, the only native Spartina species found in San Francisco Bay. It is also called Spirobolus foliosus due to a recent taxonomic revision, but we continue to call it Spartina foliosa or Foley. Otherwise, we'd be the Invasive Spirobolus project, and we're not going through that change. So this plant, it's a perennial halophytic grass, so salt tolerant grass in Poaceae family. It is rhizomatous, so it means it's got underground lateral roots like tillers and shoots, which that's how it does most of its vegetative spread. So it grows in kind of a clonal pattern. It has got fairly limited geographic distribution. It is only in California and Baja. and it is a foundational species in the low marsh. It's an early colonizer, and it's the only thing that can grow in that low elevation of marsh with the level of salinity that most of the bay has. So on this slide, in the lower left, we've got a little marsh zonation diagram. Going from left to right, or high elevation to low elevation. You've got your uplands, then you've got your upland transition zone, and then you've got your high and mid marsh, which then drops a little bit, and you've got your low marsh. This is where Spartina foliosa grows, which is denoted by that little orange arrow bar. Below that, you get your mudflats, which are by nature unvegetated. And then below that, you get into your underwater, your subtidal, thank you, your subtidal zones. So because it is the only species that grows at this level of elevation, at low elevation in highly saline water, it provides 100% of the habitat for animals, be whatever they are, that use this kind of low elevation. We have a lot of foliage in the bay. It's very widespread. 99% of all the cordgrass in the estuary is foliage. It's native, so that's great, and we want to keep it that way. Next slide. Here are the other species. So there are 4 species or types of invasive Spartina that have been introduced into the estuary. Starting with, I'm going to go through these real quick. In the upper right-hand corner, you've got Spartina Densiflora. Common name is Chilean cordgrass. As the name would sound like, it's from South America. It is a perennial bunch grass. It was brought into the estuary, oddly, as part of a restoration project in Cordova Creek. On the lower left, we've got Spartina anglica, or English cordgrass, which was also brought in on that same restoration project in Cordova Creek. The lower right-hand corner, Spartina patens, only exists in one site in Benicia, what is that, state recreation area on the Carquinez Strait. Its common name is salt meadow cordgrass. So while those 3 are in the estuary, they are very, very near eradication. We've all but gotten rid of it. Anglica, we find like 1 or 2 plants a year. We have less than a couple square meters of patens left. And Densiflora is now just reduced to little seedlings. It gets a seed bank, so it has like a 5-year seed bank. So we have to go out every year and find all those little seedlings and make sure that they don't, go to seed. So those guys are kind of on the periphery. The big problem is this hybrid, the hybrid Spartina alterniflora by foliosa. It is also, like foliosa, a rhizomatous perennial grass. And it can exist in basically any watershed that we have in the bay. It's all salt marshes, mud flats, and then all the associated waterways. Slide, please. So how did we get here? What happened? So in 1976, the Army Corps of Engineers brought in Spartina alterniflora, so East Coast smooth cordgrass, Atlantic smooth cordgrass, brought it in also as part of a tidal restoration project. They were trying to rapidly stabilize dredge spoils so that they could build up some marshes that had been degraded, had been converted to salt production ponds previously. They had the best of intentions, but had some unintentional consequences. We now have a lot better understanding of what can happen when you introduce a novel species into an ecosystem. We try to not do that so much anymore. So then sometime, we don't know exactly when, but sometime in like the early 80s, the alterniflora that had been introduced hybridized with the native foliosa and then just started running rampant and spreading very, very quickly all through the 90s. That's when people's attention started getting their attention. UC Davis did their studies And it was identified that this was really a big problem. However, it had already had generations of backcrossing. Backcrossing, basically, you've got one parent, Spartina Densiflora, another parent, Foliosa. They hybridize. You've got hybrid offspring, but then those hybrid offspring can fertilize or can pollinate with other hybrids, but they can also backcross to either of the parents. So we ended up with just a whole mess of all these different genetic forms and morphologies. The main reason it became a problem, though, is because it has these transgressive traits that allows it to occupy so many more tidal niches than any other of our native plants. Slide. So, as we were saying, we had this hybrid swarm that happened, and a lot of these offspring exhibited what's called hybrid vigor, basically where it can grow bigger and badder and stronger and just more extreme than either of the parents. So in this slide, we've got Foliosa, the native, on the left, and the hybrid on the right. The hybrid typically, not always, but it can grow taller than the Foliosa. So that means it can grow into deeper waters, much lower on the mudflat because its head is higher and it can stay above water with the higher tides. It grows denser, so it outcompetes all other plants. Pretty much nothing else persists when it moves in and takes over because it crowds it all out. And as these photos show, the inflorescences on the hybrid are much more prominent. They're typically longer, denser, so it produces more pollen, more seed, and with higher fertility. This is a big threat to Foliosa that would be maybe nearby because they get pollen swamped and then they just produce more hybrid seed. The other main thing, as I said, it can grow lower in the water. It can also grow higher. So going back to our marsh zonation diagrams, we've got the one on the left for Foley. Notice how on the right-hand side, there's the one for the hybrid. And notice how much broader that orange bar is. They can grow right up to the upland edge, and it can grow way down into the mudflat and eventually convert that mudflat to kind of elevated marsh. So where we like to say that Foliosa stays in its lane, the hybrid definitely does not. It goes wherever it wants. Slide. So how this becomes a problem with our landscapes and our ecosystems is that, yeah, it degrades the natural ecosystems or the native ecosystems by crowding out all the other plants. It will convert mudflats, which then takes away the foraging habitat for all the waterfowl and shorebirds. It also, because it grows, its roots grow very densely. And so right below the mudflat surface, you get this thick root mass that is impenetrable by the invertebrates. So it changes the food supply for all the shorebirds. Invertebrates have been shown to drastically reduce in both abundance and in diversity when the hybrid comes in and takes over a mudflat. It also, as I mentioned earlier, with the potential for pollen swamping, it can completely outcompete Pacific cordgrass, which then potentially endangers Pacific cordgrass as a species because it will physically outcompete it, but then it'll also outcompete it reproductively. And with it also filling up channels, it increases flood potential because the water has nowhere to go. It's also been shown that when the water has everywhere to go, it can pond up, kind of in the marshes, which ended up providing prime breeding habitat for mosquitoes, which then help all their public health concerns around it. So, and then the last thing, and this goes back to all of those tidal marsh restoration projects that were starting in the 70s and 80s and that are ongoing now. It's such a quick colonizer. It immediately moved into all of these sites that were opened up. This was especially visible on the Hayward shoreline with like Cogswell Marsh and Roberts Landing. Slide. Here is an example of that. It's tilted on its side. So north is actually to the right. This is looking at the Hayward shoreline. This is the mouth of San Lorenzo Creek, which is a part of Roberts Landing in San Leandro. So this just shows a little time-lapse series, a very short time-lapse series. You can see the little red box on there in 1986. This is what a few little small hybrid Spartina colonizers looked like. And by 2004, they had spread, converted all that mudflat to a very degraded, low-quality marsh, and continue to grow. If you look at that 2004 image, you can see right up at the top, there's one clone way out in open water or mudflat. And that's what it does. Left untreated, that would then completely coalesce with the shoreline, and it would just continue to convert all of this mudflat. Slide. So this is where the Invasive Spartina project comes in. We were formed in 2000 and started inventorying and surveying for the invasive around the bay in 2000. And we began full treatment in 2005. And we continue to do this on an annual basis, all during the growing season. So pretty much it's from June until about now, June to November. And our monitoring needs, how much we need to inventory has grown to about 70,000 acres of potential Spartina habitat. We try really hard to cover all of that every year. It never happens, but we do prioritize areas of higher risk and infestation on an annual basis. So how has this looked over the years? Slide, please. So here's another time lapse. So showing the distribution and abundance of hybrid Spartina over the years. We've got 4 little snapshots, 2005, 2010, 2015, and then the big one on the right is 2020. So how these are color-coded, dark green means that there is no invasive Spartina. Light means there's a very little bit. Then you get to kind of yellow, orange, and then the dark orange. Dark orange means that there is more than an acre of hybrid Spartina within that given site. So you can see through the years that we've generally transitioned from these really dense, especially in Central and South Bay, heavily infested marshes. We've really dropped that down. You might also notice that in 2020, there is a large Sassoon region that is missing from 2005, 10, and 15. Because in 2016, we found a new population of it that had established up in Sassoon. And we're rapidly taking care of it, but that also increased how much land we need to survey on an annual basis by about 10,000 acres. I'm not sure if you can see the detail on your screen, but through the years, there are actually also extra marshes that are added, especially around Eden Landing. So just on the east side of the San Mateo Bridge. These are all the efforts of the South Bay Salt Pond Restoration Projects that we work very closely with. But as we said before, pretty much every time that a new pond gets opened, it rapidly gets colonized by hybrid. And so we have to step in and make sure that doesn't happen. Slide. So how this looks graphically, here is just a graph of the amount of hybrid Spartina in the bay from the beginning until 2020. Peaked, so this is going from 2004 to 2020. You can see there was the peak at 2005, and that's when we started treatment. And it dramatically dropped, really knocked it back intensely up until about 2011. And then it's been a little bit more of a holding pattern. In 2011, we stopped treatment at a certain number of sites out of concerns for the endangered California Ridgway's rail. Lindsay will be talking about that a little bit more in a little bit. We are down to at least as of last year in 2020, we are down to 33 net acres throughout the entire estuary. I'm still doing analysis and numbers from this year's season that we just ended a couple weeks ago. And it seems like that number is going to be way down this year, which makes me very happy. You might also notice that there's a little uptick between 2016 and 2017. And that was mostly seen within these marshes that we're no longer treating, or weren't treating at the time, at least. And it was just the response from those 2 really wet winters that we had after 5 years of extreme drought. So these plants had been in these marshes, like holding on, holding tight, and then they got all the fresh water that they wanted and they just exploded. We have begun to start treating some more of those sites. So that's why these numbers are going to start going down. And Lindsay is going to talk about how we do this, like what we actually do on the ground to deal with all this. Lindsay.
Awesome, thanks, Toby. All right. There we go, okay. So, hi everyone, I'm Lindsay. I am going to be introducing you to the Invasive Spartina Project's programs. So Toby has talked a lot about why what we do is important, and I'm going to tell you a little bit more about how we do what we do. So our various programs include our inventory monitoring program, which is the mapping of invasive and hybrid Spartina. Then we have our treatment program, our treatment monitoring program, which is treating, removing hybrid Spartina. We have our Ridgeways Rail program, where we monitor populations of Ridgeways rails throughout the estuary. And we have our revegetation program, where we actually go into the marshes and add plants in areas where we've removed hybrid. Also, we go in and do the same in new marshes, just bolstering marsh habitat for rails and other species throughout the bay. So this photo is really cool to me, especially because I spend a lot of my summer in this exact area. This is Bear Island. So it's right off of Redwood City, and it's one of our biggest marshes in San Francisco Bay. And this is actually a picture of a very small part of Bear Island. And there are 2 biologists in this photo for scale. So when we go out and we do the work that we do, we at Bear get dropped off often by airboat and walk around in the marsh looking and identifying and mapping hybrid Spartina, and then going back and treating it. So looks like these 2 biologists are out doing some mapping, probably a little earlier in the season. So what does that mapping look like? We call it inventory monitoring. And our OEI biologists inventory, as Toby said, up to 70,000 acres. That's an ambitious number, but we certainly do our best. We definitely get our eyes on all of those acres every few years at the very least, but we have sites that we definitely get to every single year. So what we do when we're in these 70,000 acres of marsh is we walk around with our handheld GPS units. You can see on the left there, that's one of our seasonals from last year, Randy, and he's got his hand on his little handheld GPS. And he's looking for hybrid Spartina. It looks like he found some, that really tall stuff to the right of him. So we use them to take these GPS units to take really accurate locations for the hybrid Spartina that we find. And there are a lot of benefits for going out and mapping ahead of time before we go out and treat. And those include, it saves time. So our treatment crews don't have to visit any section of the marsh other than features that we've mapped. It reduces the impact to this sensitive habitat. So biologists can lead treatment crews right to these mapped features, which minimizes walking through the marsh and disturbing sensitive habitat more than is necessary. It allows time to thoroughly inventory the marsh and identify hybrid Spartina. So it allows us to save as much native foliose as possible, which is not an easy task all the time. As Toby indicated earlier, hybrid Spartina has many different phenotypic variations depending on marsh conditions, location in the bay, and other factors. It's also, we get the opportunity, because we do this inventory, to do some pretty extensive genetic testing every year. So we take about 500 samples every year to identify hybrid in the bay. So when we come up to a patch that we have trouble identifying, we often will take a sample, and I'm going to talk a little bit more about our genetics program later. So this is a graphic you've already seen on Toby's slide, but I just want to emphasize both monitoring extent, so get a really good idea of like what this monitoring effort looks like, as well as these widespread green and yellow sub areas. So there are 220 sub areas represented, represent these 70,000 acres of habitat. And in 2020, OEI mapped 33 net acres of invasive Spartina in those 70,000 acres. So for context, that's 0.05% of those 70,000 acres where we mapped hybrid Spartina. So as Toby said earlier, the dark green means there's no invasive Spartina. The light green is less than 10 meters squared of invasive Spartina. And to call your attention to a kind of cool area that I'm going to talk about later, the star that just popped up just to the left of it is like a little finger that's kind of sticking off. That's the Alameda Flood Control Channel. And that is the original introduction site for invasive Spartina. And it's green, which means that we've done a great job of eradicating hybrid Spartina there. And I'm going to show you some slides later of the revegetation effort that we've gone to in that area as well. So just getting a good idea of what a huge inventory effort this is. And like I said before, we do get our boots on the ground and our eyes on so much of this every year, and definitely on every acre of it every few years. So just diving a little bit deeper into these numbers really quickly, 151 of our 220 sub areas now contain under 10 meters squared of invasive Spartina, which is a huge, huge success. So just 186 meters squared, or about 0.05 acres of hybrid Spartina are left in those 151 sub areas. And I think that number is from 2020. So we're still waiting on those numbers from Toby for 2021. And the way that we actually do the mapping in the field is we do that mapping in square meters. So we go out and we spend some time at the beginning of the season calibrating with each other so that when we go out and map Spartina, we have a pretty good idea of how to look at a patch and say how big it is. Obviously, our GPS units help a lot, but we use some pretty just like, I know exactly what a meter looks like on my body. I've measured it out where like my collarbone to my fingertips is 1 meter. So if I'm kind of figuring out how big a patch is, I can use things like my body to help me do that. And also genetic sampling, like I indicated earlier, is really important to our inventory process. So OEI biologists use genetic sampling technology to help inform Spartina ID when field ID is uncertain, so that sometimes happens early in the season before plants have finished growing and haven't flowered yet. We have patches of Spartina that are lower confidence just because their phenotype is harder to distinguish. We also use it when we see new invasion at a site to confirm that what we're seeing is hybrid or foliosa. And we also use genetic sampling to create a genetic baseline for comparison. So we sample obvious hybrid and obvious foli. And we also use it to confirm native foliosa that we use in restoration planting because we don't want to get confused and plant hybrid somewhere where we're trying to revegetate with native foliosa. So if I'm walking around in a marsh and I see a little patch, maybe this is a little bright green and a little spiky, but it's early in the season and it could just be foli, and I kind of want to know because we're at a site where we're reaching eradication. I'm looking at this plant and I'm going, is it hybrid? I don't know. Maybe I should sample it. So I take my little baggie and I pull a couple leaves off the plant. If it's flowering, I'll do some measurements. I use measurements as well. And we can basically attempt to identify, like to start off by morphology, location, site history, et cetera. But if it comes down to it, we'll take a sample. So we then send our samples to genome advisors in LA, where they use 15 genetic microsatellite sequences to perform PCR analysis. This is the same basic method used for COVID testing. It amplifies the sequences of DNA markers so that we can read them. And we use the results to estimate the ancestry of an individual plant. So this graph here shows analysis of several thousand plants. That tight range and spike in the green for Spartina foliosa shows that there's a much more distinct signature for foliosa. So for the hybrid Spartina in red, the graph shows there's a much broader range of genetic diversity, which makes sense since we have this hybrid swarm created by interbreeding and back crossing of generations of hybrid plants. And we also see a variety of morphologies in the field of hybrid as well. So an analogy that our DNA specialist used when he explained this to me was, let's say Toby has a Labrador Retriever. He doesn't, but let's say he does. That's Foliosa. And then my Border Collie, Australian Shepherd, Cattle Dog, Siberian Husky, that's actually the mix of my dog, that's the hybrid. So there's a lot more going on there genetically with my dog than there is with Toby's very specified Lab Retriever dog. So then the overlapping region between the green and the red is an area where we're a little less certain. Because our goal is eradication, our threshold for what to treat is set pretty far to the left on the graph. We often kind of lump things in with hybrid when they come in a little bit uncertain. We tend to treat most of what lands in that overlapping region. But of course, the challenge is always to preserve as much Foliosa diversity as we can. So it's a little bit of a figure it out game on that front. Okay, so moving on to our treatment program. We have a lot of different tools in our toolbox for treatment. The tools in our toolbox include digging and tarping, as well as using Amazopir herbicide. So we carefully plan access and species habitat protection by going on foot in boats and airboats and trucks. And we do our inventory monitoring first, obviously, to identify and map the hybrid Spartina. And then we go in and we do our targeted treatment, which of course minimizes the impact to the habitat. And our hybrid Spartina treatment occurs between June and November, where we treat about 150 different sites. Okay, so Spartina Densiflora, let's talk about that. Okay, so we have Spartina Densiflora, Toby mentioned earlier, is one of our hybrid varieties one of our invasive plants that has become almost completely eradicated in the bay. So we do this work where we survey historical sites twice annually, mostly up in the Marin area. And we do the first one in early June when the plants are flowering, which helps in detection. And we do our second in January when the native pickleweed has senesced. So it's brown, which makes the plants a little bit easier to see. All of our plants are manually removed now and deposited off-site. And that's because, as Toby mentioned, we really are only looking at seedlings at this point. But it's really important to find them because that seed bank is 5 years. So we go out and it's really like finding a needle in a haystack, and we really try hard to find those little seedlings. We haven't needed to use herbicide for Densiflora eradication since 2012. And our status as of 2020 was we found 66 seedlings, which totaled 5.6 meters squared. And that represents, over the course of this project, a 99.9% reduction from the peak. Okay, so to talk about Amazopir herbicide, that's how we treat hybrid Spartina. And we use this herbicide in most areas of the bay. The EPA considers Amazopir a category 4 practically non-toxic to wildlife, including mammals, birds, fish, and aquatic invertebrates. And ISP has done water quality monitoring that's detected concentrations in the water after treatment that are far lower than any amount that would be of concern for wildlife. There's a 90% reduction detected within the first week as Amazopir is broken down by UV sunlight and has a low potential to bioaccumulate. And this treatment takes place with the close monitoring of trained biologists. And we also stop treatment in an area if we detect an endangered species, especially if we hear a ridgeways rail nearby. And that's not really because the Amazopir would be harmful to them. It's because we don't want to disturb them in general. So when we're out doing inventory or putting plants in the ground. We're very conscious of what we hear and what we see around us to make sure that we're not disturbing any endangered species. So the Invasive Spartina project is really an example of a conservation dilemma. So we have endangered species versus invasive species. And human development has led to this massive loss of tidal wetlands in San Francisco Bay, with wildlife that are dependent on wetlands now becoming endangered species. And then human action then led to this introduction of a highly invasive species, which in the case of hybrid Spartina does help create some type of habitat. But how do we balance removing invasive Spartina while protecting endangered species? And this is also taking into account the fact that while hybrid Spartina is creating habitat for ridgeways rails, it's also removing mudflat habitat for all sorts of shorebirds. So there's so many different factors at play here. So what is the answer? It's adaptive management. It's treatment, restoration, and then surveys to assess populations, and then reconsultation for further treatment as we move forward. So, like I mentioned, hybrid Spartina benefits ridgeways rails. It creates brand new marsh where there was none before by invading mudflats and invading new restoration sites. And it grows really tall, so it provides great cover at high tide. It also outcompetes native Spartina and other native vegetation. So the removal of invasive Spartina creates this temporary deficit in ecosystem function, the function that had been temporarily created by the hybrid that's usually provided by the native plants in the native ecosystem. So how do we keep track of the rail population to know if our restoration efforts are impacting this species? We conduct call count surveys between the months of January and April, which is the start of the breeding season. And we do this by collecting, there are data collection points that are located approximately 200 meters apart along a transect that we've designated. And biologists stand at each point for 10 minutes and broadcast different bird calls. We do a black rail and a ridgeways rail call at minutes 6 and 7. And then our survey results represent just a subset of the population at the site. So not all birds will call, about 60% detection probability according to one USGS study. And then biologists record the distance and location of each call and physically map the birds after the survey by plotting them out on a piece of paper manually in order to account for repeat birds, because you can hear the same bird calling from more than one station. So despite the fact that we're removing habitat, rail call counts over the last 10 years have increased. The data here was collected by ISP and other partner organizations, including USFWS and Point Blue. And this is really due in part to the successful revegetation program that helps to mitigate the impacts of removing invasive Spartina. So it all comes down to this cute bird, the endangered ridgeways rail, and it's really the center of our revegetation program. Although we've mentioned other species that are important, we're very ridgeways rail focused when we work on re-veg and also going out and doing our rail call surveys. So one of our main challenges is obviously balancing hybrid Spartina removal with the need to protect habitat for this animal. So ISP's restoration program really grew out of that challenge. And our main goal right off the bat was to rapidly enhance tidal marsh habitat to benefit ridgeways rails primarily, but also other tidal marsh dependent wildlife. And the focus of our program has been at enhancements at specifically invasive Spartina project treatment sites. So ISP's, the Invasive Spartina Project's re-veg program was started in 2011, and we just completed our 10th year of habitat enhancements. This program is guided by our plan, which is formed by our tech committee and implemented at a bay-wide scale. The program focuses on critical components of rail habitat, so that's cover from predators for foraging, roosting, nesting, and high tide refuge. We actively use adaptive management to learn from enhancement efforts and inform future efforts. For example, in the first year of our program, we lost most of our native cordgrass plantings at one site due to grazing by Canadian geese. So we experimented with rope caging, and now at new sites, we initially rope some of the sections off to protect at least half of the native cordgrass plantings until we can get an idea of the likelihood of grazing at that site. So this map here shows the 40+ sites where we've implemented habitat enhancements throughout the San Francisco Bay. The blue areas on the map show plantings, primarily planted Pacific cordgrass, Spartina foliosa, and marsh gum plant, Grindelia stricta. And I have some pictures of Grindelia coming up. So the orange shows where high tide refuge islands have been constructed. So those are like mounds that actually we build up in the marsh and plant gum plant on top of, and those create habitat at high tide. It's actually that time of year right now. So one of my coworkers was out yesterday monitoring while a group of our contractors was building a high tide refuge island over, I think right on the east side of the San Mateo Bridge. Yeah, so let's see. What have I not said? This photo here at the bottom shows our ISP staff and contractors planting gum plant along a channel to provide refuge for rails. So that's one of our planting days down there. And you can see that there's the little pink flag is when we go out and we actually do ground truthing. So biologists go out and determine the best place to put plants in the marsh. And then we come back out with our contractors later, and there's like a planting design we have. And that determines where each of those little marsh gum plants gets plopped down in the mud. Okay, so in most tidal marshes of SF Bay, the vertical cover is provided by a pretty small suite of plant species, primarily Spartina foliosa at lower elevations and Grindelia stricta along the banks of tidal channels and in mid and high marsh at those higher elevations. So our program focuses on active planting of these two species specifically. And this slide shows examples of a few sites where we have done this planting. So here on the left, up on the top, we have Pacific cordgrass, Spartina foliosa, planted out at Eden Landing, which is right on the east side of the San Mateo Bridge. And this was an early restoration project. So it was mudflat before, and then you can see 4 years later, this healthy band of Spartina foliosa along the channel. And then down at the bottom there is a picture from AFCC, the Alameda Flood Control Channel, which was that little finger that I pointed out earlier where hybrid Spartina was first introduced. So that is a happy band of foli. We went in and we removed hybrid Spartina and we brought foliosa in, and it looks fantastic. And on the right is two examples of Grindelia stricta being a gum plant being planted along two different channels, I think both along the Hayward shoreline. And after this restoration, the pickleweed came in, but we didn't get any recruit of Grindelia stricta, so we went in and planted there ourselves. And this cover's really great for ridge-raised rails, and we had great successful planting along each of those channels shown. Within 4 and 5 years, we just have this huge healthy band of gum plant there, which is such great rail habitat. So here's a great example from Eden Landing. This is the Eden Landing Ecological Reserve, and this photo shows the site back in 2011. It's just an open, unvegetated mudflat after the opening of the tide gates and the restoration of tidal flow. And this site is pretty far from any local source of native cordgrass, which would be the marsh plant we would expect to colonize given the marsh plain elevations here. So we wanted to jumpstart the trajectory of this site from open mudflat to potential habitat for RERA, for ridge-raised rails. So we did some active planting of native cordgrass here. So this slide shows, first of all, on the right, sort of an example of what I get to look at on a sort of daily basis, depending on what my day is looking like that day. But this is like an aerial view of part of Eden Landing. And everything you see in green is foliosa plantings, and everything you see in orange, just along a few of those channels, it's a little bit more faint, that's Grindelia or marsh gum plant plantings. The other thing you'll see is photo points. So the letters A, B, and C are all corresponding to those photos that you see on the left. So there's photo point A, photo point B, and photo point C. Those are locations that we go to every year to take the same photo, so we get an idea of what things are looking like and how successful our restoration efforts are. So you'll see that in photo points A, B, and C, those are all examples of mudflat to healthy foliosa. And then last thing you'll see on this map here is rail detections. So when we go out and we do our Ridgeways rail monitoring, those rail detections, the locations of them get entered into this too. So when we're out there doing our work, we have an idea of where rails have been heard earlier in the year, which is really cool. So here's what the site looks like now, from mudflat to marsh. And the Ridgeways rail population at this site is responding by rapidly expanding. So we had no Ridgeways rail detections in 2018. We detected 8 Ridgeways rail in 2020, and we detected 13 Ridgeways rail in 2021. So things are looking really good over there. So here's an example of the rapid spread of invasive Spartina, and then we're going to show some successful control and then restoration with planting. So this sort of takes us from beginning to end, this next series of slides. So note in this slide, the size and position of the clones circled in red and blue. They're just different colors kind of to help you keep your eye on them, and their relationship to the rest of the hybrid and the shoreline. I don't think I said this. This is the Alameda Flood Control Channel. So again, the original introduction site of invasive Spartina. So you'll see here that the red circled clones, which were leading the edge, winning the race back in 2000, are lost in the hybrid Spartina meadow 5 years later. And the blue circled clone, which was previously isolated way out on the mudflat, has grown considerably and is now being assimilated into the shoreline meadow. So we've lost all of that mudflat completely. And here's AFCC now. So a few more great pictures. You'll see the one on the top left has a happy band of foliage all along the edge, leading up to this great stretch of mudflat that gets exposed at low tide. And then obviously the channel itself, which is clear and can flow and do all the things that it's supposed to do as a channel. And then some more pictures of just great habitat restoration. So mud on one side and beautiful foliage bands on the other. One thing I will point out, I don't know how interesting this is, but those poles you see in those photos also help us position our photos every time we go back. So we actually have little PVC pipes that we leave out planted in the mud that help us find where the photo point pole goes and where we're supposed to stand, so we can get the most accurate photo year after year. I don't know if any of you have ever tried to take the same photo in the exact same place multiple times. It's really hard. So with that, I'll just say that the Invasive Spartina Project is, I think, as we've indicated, this constantly moving target as we continue to remove invasive Spartina, restore native marsh habitat, and monitor the California Ridgeways rail and other marsh species. And we hope that working toward this balance can help create and protect healthy native marshes that are more resilient in the face of sea level rise and climate change. So with that, I'd like to thank all of you for listening to Toby and I, and we'd be so happy to take your questions.
Wow, that was incredible. I knew nothing about marshes. This is such a wonderful overview. Thank you so much for talking us through how we got this invasive species and what you've been doing to not only get rid of it, but to monitor how you get rid of it and how it benefits the ecosystem and our endangered birds. So we do have some questions. Susan Malini asks, the Spartina removal started in 2005 and the Ridgeways rail population plummeted in 2007. How does the current population compare to the pre-project number of birds?
Gotcha. I don't know the specific numbers offhand. Yeah, 2008, we definitely saw a big dive in rail populations, both in North Bay, which didn't have any hybrid Spartina treatment, but then also in South Bay where there was more. I am pretty sure that our numbers are well above 2008 levels currently. Definitely had a big dip in 2008, but then we've been etching back up every year. Does that answer your question, Susan?
I think they wanted to, there's an awful lot of Susans here. So
oh, sorry, Susan Mulaney, I think you said her name was.
Yeah. So I thought she wanted to know, when I read this question, do you have any information about how many of the ridgeways rail there were prior to the invasive species arriving?
Oh, no. There was rails were not monitored. Rails were not surveyed for before the invasive Spartina project started. That was really the big springboard to start inventory of rails. So no, we don't have much of a baseline number. There is an estimate that in the early 90s, there were only about 300 pair available, about 300 pair in the entire bay. That was, there was plenty of hybrid. There were also a lot of red foxes around that were really knocking down the rail population. But no, prior to 2005, there were not any consistent rail surveys. So we don't really have before then, unfortunately.
And I'm sorry, I don't think I understood. A red what was knocking down the rail?
Red foxes.
Oh.
Also an invasive species. Red foxes were introduced in South Bay. We're still dealing with them, but their population was a lot higher than it is now. And they were preying on rails a lot.
Oh, do we know why they were introduced?
No. No, we do not. We just know that they're here and they're still doing their thing.
Okay. And have the rails moved into any of the sites where, again, from Susan Mulaney, where the hybrid Spartina has been removed? Are the rails using any of the gum plantings?
Yes, very much so. So we definitely have great examples. Say an example, Cogswell Marsh A in Hayward. There was completely overrun. It was a monoculture of hybrid. We got rid of the hybrid. Numbers of rails temporarily went down, but then we also planted a lot of Grandelia, and rails are back there in the highest numbers that we've seen. There are also, I think Lindsay referred to this in her portion about North Creek Marsh in Eden Landing. That also had a lot of hybrid in it. We got rid of the hybrid, and then we planted a bunch of Foliosa and Grandelia, and rails have been increasing on an annual basis. Similarly, Sonoma Baylands had hybrid, and we got rid of it, and now there's about 30 rails detected there every year, where prior to getting rid of the hybrids, there were no rails.
Wow.
So yeah, we have a lot of evidence that rails are responding really well to our restoration efforts.
So Susan Mulaney was more specific. What are the numbers of rails from 2005 to 2007? You said that counting started when the project started?
I don't know those numbers offhand. Sorry. It should be available possibly on our website, though. These are public documents. We do annual reports. So this should all be available online.
Sounds great. Yeah, it's wonderful to be able to see the connection between the ecosystem restoration and the wildlife that use a specific plant and ecosystem.
Susan, may I ask a question? This is Bob Hall, board member of your Buena chapter. I'm fascinated by this presentation too. I've learned so much, and thanks for showing us the connection to how important the mudflats are to multiple species. I know, like, managing for one individual species is tough, but you also got to consider the multiple mudflat species, which may not be endangered, but they may be declining, and we need to offer a room for them as well to eat. So I'm wondering, does the Spartina foliosa, the native plant, does that play well with the gum plant? It sounds like they play well together. And when the hybrid crowds out species, can you name some of the hybrid, some of the species that crowds out?
Sure. Yes, Spartina foliosa and Grindelia stricta play very well together. They're great complements to each other because they grow at different elevations in the marsh. So the Spartina foliosa will grow in the low marsh, so on the bay fringes and down in the channels and on the benches of channels, whereas the Grindelia is going to grow up on the marsh plain, lining the channels. So it provides a really good transition for rails to stay hidden from predators in the Spartina down low, and then as they jump up to the marsh plain where they nest. So yeah, they both, that's why we focus on these two, because they're both very beneficial, especially for rails, but also for salt marsh harvest mice and black rails and other sensitive species in the marsh. And I think there was another question in there.
Yeah,
as
I was
well.
asking if there are any other species that you can name that it does crowd out, that we need these species because maybe they serve specialist species. Some other, you know, we're finding out more and more how important to bees some specialist plants are, and without those plants, then you know, you're getting rid of those species as well. So, are there any specialist species out there that are being crowded out by this hybrid?
Yeah, I'm not sure if we know. I could definitely name off other plants that get crowded out. We don't know of any of these kind of specialist benefits that they might provide. But the hybrid Spartina will crowd out the Grindelia stricta, pickleweed, so Salicornia, virginica, what else? California sea lavender, Limonium californicum.
Is Marsh Baccharis? Is Marsh Baccharis out there?
Marsh Baccharis, yes, they're not so much in direct competition. We don't have all that much Marsh Baccharis in the bays. They tend to be a little bit more in the brackish zone, whereas most of our hybrid stuff is much more in the hypersaline zone. But it would crowd them out for sure where they exist. It'll crowd out everything else, basically.
Okay, one final question for me. We're very interested in relationships between Lepidoptera and plants, and we're asking a lot of questions about what can eat what. Do you know anything about what can eat the native plant, Lepidoptera-wise, if anything does, and can it also eat the hybrid or the original invasive as well?
I have no idea.
Okay.
Lindsay, do you have any information to that? Lindsay, are you there?
I'm here. Yeah, no, I don't know of. That's a great question.
Sounds like a really good grad student project.
Yeah. All right. Thank you. Just a fabulous presentation. Thanks so much.
And we do have a great comment from George saying that the red fox was introduced into lowland California in 1880 for fur farming and hunting.
Oh, that makes sense. Yeah. Yeah, we definitely had a big movement of them in the Bay Area, a big expansion of them in the Bay Area through the 80s and 90s. And so they started doing active control.
And according to CalScape, there's at least one moth associated with Spartina foliosa, and that's the small engrailed. It looks pretty. And I know that pickleweed is associated with one of our rare butterflies. So.
Okay.
Well,
I would imagine that Limonium californicum, it seems like they might have some associations as well. They're very, very floristic.
And Claire Myler in the chat has added the 2020 Ridgeways Rail Report, which may have some historical data for reference in terms of numbers of birds. So that information is available in the chat. And if you want to save the chat, you can go all the way down to the bottom of the Zoom chat. Bottom right-hand corner, there's 3 little dots, and it gives you a chance to save the chat, so you'll have that. Let's see if there are any more questions. All right. And I do want to go ahead and mention this. Susan Linney had asked who Saved the Bay was founded by, the name of the three women in it: Sylvia McLaughlin, Esther Glick, and Kay Kerr. So lots of good questions, lots of, it's always so much fun to work on a CMPS presentation. There are so many great people with such varied backgrounds and interests. All right. If there's no other questions, I want to thank you both so much for a wonderful, informative, just beautiful presentation. I learned so much.
Awesome. Thank you for having us. It's been fun.
Thank you.
Good work. Thank you so much. All right. Goodbye, everybody, and see you January 13th, 2022 for the Cynthia Powell Cal Flora presentation. Good night.
Good night.